Patients with irritable bowel syndrome and hypermobile Ehlers-Danlos syndrome demonstrate an altered colonic neuroinflammatory profile when compared to those with just irritable bowel syndrome alone. Hypermobile Ehlers-Danlos syndrome is characterized by tissue fragility, joint hypermobility, and skin hyperextensibility. Hypermobile Ehlers-Danlos syndrome is a multisystemic disorder associated with several comorbidities, including features consistent with IBS. In IBS, abdominal pain is associated with heightened colonic sensitivity, allied to an increased density of nerve fibers and increased expression of mast cells. Animal studies in Tenecin X knockout mice, which exhibit features similar to patients with hypermobile Ehlers-Danlos syndrome, have similarly demonstrated an increased expression of nociceptive CGRP nerve fibers in the colonic mucosa. In this study, we characterized the molecular changes in the colonic mucosa associated with abdominal pain in IBS alone and in comorbid IBS and hypermobile Ehlers-Danlos syndrome. We assessed for the presence of nerve fiber density using the markers CGRP and PGP 9.5, and also for mast cell expression using mast cell tryptase. Biopsies were obtained from the rectosigmoid colon of 4 patients with IBS alone and 4 patients with overlap IBS and hypermobile Ehlers-Danlos syndrome. The biopsy samples were stained with CGRP, PGP 9.5, and mast cell tryptase. Mast cells were counted using the multi-point tool on ImageJ, and nerve fiber density was assessed using pixel analysis via ImageJ. Statistical significance was tested by using an unpaired t-test. We demonstrated in this study that in the human colon, the overlap IBS and hypermobile Ehlers-Danlos syndrome group demonstrate a 2-fold proliferation in PGP 9.5 immunoactive fibers compared to those with IBS alone. However, this is not the case for CGRP positive fibers or mast cells, which we demonstrated to be significantly decreased in our overlap IBS and hypermobile Ehlers-Danlos syndrome group. In contrast to animal models of hypermobility, these initial results demonstrate that CGRP is very limited in the human colon in patients with IBS and overlap hypermobile Ehlers-Danlos syndrome. The two groups demonstrate a distinct difference in neuronal innervation of the gut, with the IBS alone cohort displaying an increase in the expression of specific nociceptive fibers and inflammatory markers. This suggests that different inflammatory and nociceptive pathways may be involved in patients with comorbid IBS and hypermobile Ehlers-Danlos syndrome. This signifies that the overlap group may form its own subgroup within a wider IBS cohort. However, this requires further investigation in larger numbers of patients. Ultimately, this data may enable more targeted treatment of IBS symptoms in hypermobile Ehlers-Danlos syndrome versus IBS alone in the future.